Good afternoon, everybody. So my name's Menna Clatworthy. I'm based at the University of Cambridge, and my co-lead for this session is Anna Greka. So what I'm going to do, this is the kidney focus group, and what I'm going to do to start with, I thought it would be useful for us to review what data has actually been submitted to the DCP of the HCA from a kidney point of view. And then we actually put together a variety of topics for discussion. So really we want the most of this session to be you interacting, or us all interacting, to talk about some of the challenges we're facing and potentially some solution for those. So this is a summary data slide of how many samples have been collected in terms of number of individuals that have been sampled. And you can see that it's divided into mouse and human. So if we focus on the human side of things, so the kidney is actually the blue bar there that has 22 individuals that are sampled in which the data has been submitted and uploaded. So we're doing relatively well. So the biggest samples set that's there is actually from blood, which is obviously a lot easier to get than kidney. And this divides it per organ system, which shows something similar there. Now, one thing, and I think this highlights something that maybe we should come back to. So when I asked the DCP what information they had about where precisely in the kidney this data has come from, you can see for most of the samples, the information on the precise anatomical location of the sample wasn't provided. So for some we do have information, and these are most likely, of course, to be cortex, because that's the thing that's generally sampled and is easiest to get at. But we do have some information on medullary samples, and then also pelvis and ureter too, but that's something for us to think about going forward. And then the actual methodology that was used to generate the data. So you can see that this is principally 10x data with a little bit of in-drop and SmartSeq too, but it seems to be mostly to generate volume of data that as a community we're generally using the 10x platform. The projects that are registered, so this is just in terms of number of projects that are registered rather than contribution of cells or samples, so they're focused, I guess, on some US centres and UK centres. So that gives us an idea of where we're at, and of course there's a lot more data out there that perhaps hasn't yet been uploaded to the DCP, I would say, and that's something for us to think about. And then in terms of topics for discussion, we've divided them into sort of technical and legal issues, methodological and scientific issues, and then potentially things related to how we're working to collaborate. So Anna, should I hand over to you to maybe start going through those?
Sure. Yep. Hi everyone, I'm Anna Greka. I'm at Harvard Medical School at Brigham and Women's Hospital in the Broad. So it's great to be here, and let's dive right into this. Hopefully have a discussion. We were just discussing with Matthias the legal aspects of MTAs and kind of being able to share tissue around to different sites, and so that's actually maybe a good place to start our discussions. It seems that this is easier said than done, and so we wanted to, really dive into that. So that actually affects how we're doing on tissue acquisition in terms of actually being able to share it and being able to apply multiple methods. That actually affects the working group, the standards working group. Wolfgang, I think, is the representative of that group here. And so are there legal hurdles, also, I suppose, additional ethical hurdles that we need to overcome. And then the question of ethnic representation as well. Maybe we'll stop there instead of going through the laundry list of questions and see if people have comments on those subjects of collecting with enough representation across ethnicity and other things, and then also issues around sharing maybe what you know about your own institutions. I don't know, Matthias, if you want to start. You have a lot of knowledge in this area. Thanks, Ben.
Hello, it is on, sorry. Matthias Kreister, University of Michigan, and yes, we have had several frameworks where data and sample sharing strategies had to be developed and implemented, and certainly the landscape has gotten significantly more challenging over the last couple of years for sample sharing across the Atlantic. There are two principal strategies. One, that bilateral agreements are established between entities. Some of them might already be in place and could be leveraged, or at least the language transfer to those. The alternative is that other large networks have obviously addressed that problem in the past, and taking language which was successfully implemented and accepted by institutions represented in that network as a joint framework to move forward. That is facilitated if there's a significant stick attached to it, in the sense that state-level entities are endorsing these approaches, which is one of the differences in HCA, that we don't have that, which gives us a lot of flexibility in other terms, but here it might be one aspect which is missing. But certainly in the framework of the Kidney Precision Medicine project, which is diseased kidney analysis with some smaller side project to also work on the healthy kidney side, we have a standardized framework in place which over a dozen North American institutions have agreed on, and that might be a starting point to see if this would be acceptable to the legal entities who have not signed up to it. And we have done in this framework already three pilot studies where tissue was shared among these institutions, undergone various analysis and data brought back to a central place. So that has been somewhat pressure tested already.
So I guess the sharing of samples becomes particularly relevant to this seed network around comparing different protocols. And so maybe making those agreements available so that we can look at those. Because I think when we talked about sharing samples, say from the UK with US centres to try out or compare different methodologies, the idea was to just form bilateral agreements. But I think ultimately in the long term, having one agreement that was for HCA work that each of the institutions could just buy into would make it far more flexible in the long term.
Yeah. And actually there is even an added layer here, is that with the initial QC method pilot study in the KPMP network over the last 2 years being really helpful in benchmarking and comparing data sets. At our meeting a couple of weeks ago, we actually wanted to bring to this group the suggestion that maybe there's even a possibility to have this type of reference tissues available not only for the interrogation sites, research labs in HCA, but also have the same tissue available for technologies utilized in some of the parallel networks with slightly different tasks, like HubMap, KPMP, and in the kidney space, RBK. And this is a suggestion which I would like to bring to this table. If this is something the group would feel comfortable, that we have one tissue source where we have 60, 100 aliquots preferentially generated in a way each of the different technologies are requiring that, and then have a joint reference point available even beyond human cell atlas. And certainly our experience is that it's invaluable to have this common source when you start to get into the problems we heard at the last call at the plenary session.
So if we were to make this extremely concrete, where does tissue need to go? Like, what are the specific places? So in your group it would be Barcelona, yeah, and then Germany? Japan.
Now let's see if I get this together. So yeah, basically Japan, Boston from Cambridge,
Yeah.
and Cambridge to Boston. Barcelona, Belgium, Riken. That's it, I think. Yeah.
Okay.
And I mean, that's of course There's,
It's a lot,
I mean,
yeah.
yeah.
What's the situation in Japan? Do you know if you can accept or send out tissue here? Ben is making his way.
Yeah, I think is work in progress, and the discussion how to to match the the agreement. I'm gonna have a meeting with the ethical committee in the mid November, and by that I think we may be cleared to receive samples started of that experiment.
Sounds hopeful.
By the end of November probably we may be approved.
But I think taking some research contracts that are already in place to use as a template might well be useful for these things. So.
The existing framework, Matthias, that you have now is only for U.S. sites. Is that correct? Or are there any non-U.S. sites in that agreement?
That was US sites only.
Only,
Yes.
okay. Yeah, I don't know, the complexity, to me, I mean, I'm not a lawyer, obviously, but it sounds like the complexity would increase if you're going beyond. But nevertheless, you know, maybe we should push for that so that, you know, it's like laying a foundation for the future, like you were saying, because we will inevitably need to do that. The other thing that I took from what you were saying, Matthias, was about the interactions with other groups who are doing single cell work in kidney in general. And for both for benchmarking or for looking at standards across different methods, but also potentially just for overall increasing our samples analyzed. And so how would we go about that? I know you also represent those other groups, so can you talk about that?
No, I certainly represent my opinion. Yeah. And these groups have governance bodies where that would have to be discussed, but we had, you know, from investigator to investigator discussions that everybody agreed that finding ways to generate data in a way that it can subsequently jointly analyzed, I think is a mandate we all feel we are under. Yeah. And
so we have KPMP and rebuilding a kidney.
And we have HubMap, where, you know, there is a HubMap-wide effort, I think, underway to find a way how HubMap should talk to HCA, but I think we certainly should pursue that in parallel. And I think Blue from UCSD, who is part of both of the networks, is in the room and can maybe speak to some of that effort, or if you have some comments. And I think it really would be wasteful if we miss the opportunity right now that if we identify what principal metadata standards we need to capture, and if we all agree that having a reference sampling point in place to get us into a position that down the road the data can talk to each other.
I think on the metadata front, so the DCP have produced a minimum set requirement. So I did ask Laura for that. So I think that's kind of in place.
That I think would be, and then I will stop. I don't want to monopolize the discussions here. Sorry about that. But KPMP has done the same, and I think the next step really should be that these meta standards are compared. And it might be helpful for both of the networks actually to see what blind spots each of those have right now so that we can enhance the standards. They obviously will be somewhat technology specific as well, but certainly on the sample acquisition sides, hopefully there will be a key overlap.
Any comments? No. Well, does that imply, I mean, very concrete, should we have a joint meeting then? Do we need to organize that for kidney? Does it need to be an in-person joint meeting with the other groups to really talk about these things? That's...
The next step should be that, Blue and Sanjay can take it up in HubMap, certainly in KPMP. I can take that forward, and if this group agrees that this is something HCA wants to pursue, then we should find a venue to coordinate. If this has to be an in-person meeting or can be a series of conference calls, we can decide what is the most effective way. but I certainly would love to see that happening. Yeah.
Yeah, I mean, I think it makes sense to gather data wherever it is into one common platform as much as is possible, so that everybody can use it. And you know, we heard about the more cells we have, the more likely we are to identify rare subsets, different cell states. You know,
Even if we don't reach that stage that it all ends up in one place, it at least has to be generated in a way that it can be compared. And I think that is, I feel, the minimum. And with all of these activities starting off right now, we are at a place that we can at least achieve that one. Yeah.
On the very practical side, I mean, are there SOPs for how to collect the DNA? And because I think that's really where where it in the end might fail, right? So you collect kidney, and there are many, I mean, if I think about our projects with all the different requirements and sortings that you need to do on one sample, that's already hard enough. So I think practically it will only work if you say, okay, here's this consortium, this is kind of the type of protocol I need in order to get some cells. I mean, DNA is easy, of course, but yeah, I think on this level I see, additional to the legal sides, on this level I see the biggest, because if that doesn't happen, then it's gone, right? I mean, so I think that would be another part of practicalities that need to be shared. It's not only, would like to save some HubMap samples, but actually really how.
And that's, I think, where the protocol IO system platform, I think, is a very effective one. And in the development in KPMP, the protocols are currently publicly available of how the material is procured, and we will upload into protocol IO so that information is available, and that already has been benchmarked across multiple tissue interrogation sites, and it was very helpful to see, if you have the same tissue source, to see strengths and weaknesses of the different technologies. And as we saw this afternoon, it actually helps to build a full picture if one can benchmark that against a single reference unit. So I think that's an aspect where I certainly can state for KPMP, and Blue, again, please speak up, that these protocols are already publicly available, how to procure the samples, and they have been actually specifically developed to work on biopsy material. So they are scaled down to work out of 3 biopsy cores. Obviously not with the same technologies as I think HCA will pursue, but at least with one set of technologies already.
Yeah, I mean, there's a lot of protocols in protocols IO, but I guess the question of a consensus protocol is, I guess, what you're looking for, Wolfgang, right? For every, like one perfect or agreed upon SOP that everyone uses. It's hard.
I mean, I think. And not just that, so predominantly as we saw, the samples come from cortex, which are relatively easier to process. Actually, when you're trying to take medullary or pelvic samples or even ureter, those just the really dull tissue disaggregation protocol has to be different, or you just don't get cells out. So there are some nuances around that too. And I suppose if we again, practically, to get down to the hard nuts and bolts of it, if we're saying that, say, for cortical samples, we should have a common methodology, then should this group decide what that is, or to try and put that framework in place? Because, as you say, there are many protocols already on protocols.io. I don't know.
I mean, practically, I think it will be very difficult or even impossible at this stage to have SOPs across different sites, and even if you have SOPs, then the practicalities will still differ so much. I mean, to some extent it's also worth to embrace this diversity in this first period, because this diversity will also tell us something about robustness, and then maybe in the second phase it will converge. I guess what I just meant with collaborating with other entities or other groups, and you make more out of a single sample, and I mean, that will happen on I mean, some samples might be too small, right? You will have a priority list, and it's about getting better, not necessarily optimal. I think just the SOP, if you want a sample, then what I just want to stress, right, then the most important thing is to get a protocol how to do it, because then at the site where it's collected, this will decide whether it happens or not. I think this is just, I don't think that we would need necessarily at this point SOPs across the different sites. I mean, it will anyway evolve, and people will talk and it will converge, but I do not see that we will get to an SOP level that is really realistic at this point in time. But yeah, I mean.
Other thoughts on, I guess we're still talking about standardizing. Yes, Matthias.
But then you have to be aware that you will have a significant confounder who will limit, if not make it impossible to compare data sets, particularly in the kidney when you have different ischemia exposures, different processing artifacts in technologies when we cannot benchmark them against us. If you have a reference sample where these technologies have been run against, then this can work. But if we have non-standardized protocols with incomplete metadata and no shared reference sample, then these data sets will live in isolation. If you get congruence of data, that might be informative. Discongruency in data will not.
Yeah. I mean, like for you, do you have to use the same protocols if you prepare your kidneys? I mean,
I don't know. I think that roughly speaking, I mean, we understand what cortex, medulla, and pelvis mean when we are, taking biopsies, at least position. Yeah. Now, in terms of every human kidney is a little different from every other. So, one nephrectomy is different from another nephrectomy, and as far as the actual protocols of dissociating cells, I think they're roughly similar, or if not exactly the same. But it's more about the origin of the tissue, and honestly, like how long it took from the piece of tissue to make it from the operating room. Like in my hospital, they have this thing where it goes to the special dissection room, and then a pathologist takes out the piece of tissue and then hands it to us. I don't know, every place is a little different. So there's a factor of time that is not totally controlled, each case. I mean, that's sort of been our experience so far.
Yeah, I think broadly it's true. We all have a mechanical dissociation point and then an enzymatic digestion point, which varies in the length of time and the enzymes used. But then, as you say, there are differences in terms of the warm ischemic time. So.
That's significant because that will impact the survivability differences.
Yeah. But I think that we probably have people. It comes back to what the sample source is. We have people that are using kidneys from different sources, so tumor nephrectomies versus discarded transplants. So each of them have their pros and their cons. So and then there are disease biopsies which are obtained immediately.
I think one additional source is now delivering donor protocol biopsies. This will be very scarce, but in our hands, using bulk data in the past, we could show that those clearly the most valuable reference material because they are least artifact prone. But we are talking about 1 or 2 cores available, so these are ideal reference material after optimization of protocol has been performed on the other starting points.
But exactly, those have some advantages, disadvantages. You can't get, well, it depends on who's doing the biopsy, but you can't get the pelvis and
you don't want
ureter. So, yeah. Okay, so maybe one of the things we said we'd talk about was getting broad ethnic representation. So, I mean, definitely one, from our point of view, and in my centre, the samples that we have access to are predominantly Caucasian. So, Anna, what about you?
Yeah, we already have, even though they're not in the DCP yet, but we are already in the process of collecting samples that are non-Caucasian. And I know that we have also discussed that the additional samples that we're looking to get also through Michigan and potentially through the Bay Area, there may be some additional ethnic diversity there. So hopefully we'll do a little bit better with that as we move forward. But that's always a concern. The vast majority of GWAS data has been generated in people of European ancestry, so we can plan for that not to be the case here, but we have to work on it. I don't know if there's anything that you want to say about H3 Africa and.
So these are deceased biopsy, also indication biopsies, which are procured with a protocol which has been used widely in Europe, North America, and Southeast Asia, and where now 640 biopsies have been procured in RNA later, in essence. And these have various underlying diseases, but nevertheless could offer us an opportunity when we are detecting signals in the main activity here, and we can develop deconvolution strategies to see if these signals are present in these biopsies. Again, it will be overwhelmingly cortex, but with a good representation of the nephron in the medullary rays as well. So that's an option where these data are available. They are generated with NIDDK funding, so the clear expectations that they will be publicly available and open in future. But again, this is still at the procurement phase. Yeah.
Any other comments on ethnic diversity samples? Yeah, Wolfgang.
Is there any, I mean, ethnicity is anyway a different concept coming from evolutionary biology. There will be anyway gradients. Is there a plan to, for sure, have some materials that at least would do SNP arrays? Because in the end, we'll not talk about ethnicity, but kind of geographical gradients, and then we would need to map that using SNPs, which would be almost a no-brainer, but maybe this is not so much on the radar of many. But I think the only thing it would take to cover ethnicity is SNPs and map it on a geographical scale, because there are no discrete differences, right? And then after all, one should be reminded that, I mean, 80% of the genetic variation is shared across the world, right? And then only a small fraction. That can be, of course, rare population or region-specific variation, but until we get to that, right, we talk about numbers that are way, way, way higher, right? I mean, then we are into GWAS domains and rare variants, and I think we're far from that. So I think one practical solution to the ethnicity diversity thing is that one should be reminded that genetically we're more identical than different. And secondly, that it's not groups, but geography, and then we should use SNP panels in order to map this parameter basically on a two-dimensional scale, rather than collecting Caucasian, Asian terminologies, which are always, to some extent, also difficult.
So I think, yeah, if you set out to sample individuals that you think have a different genetic background, albeit I take it that there's a lot of similarity, then inevitably, of course, there will be things that you misclassify, and genetically it's obvious that they're not, say, of European ancestry. But on the other hand, I think we do have to be careful that we go out to not just, I think the danger is that we just sample individuals of European origin. And I think from a disease perspective, so as a nephrologist, there are we are all 80% similar, but there are a number of kidney diseases that definitely have a much higher frequency, so in those of non-European ancestries, and also the diseases have a much more severe course, so autoimmune diseases like lupus and lupus nephritis, even diabetic nephropathy, so I think that is important. It may be at this stage that we're so early on in the process that we don't need to worry so much about that, but ultimately I think that does have to come in our remit as we think about disease. Did you have a comment?
You have to remember that it's not a scientific question. It's a political question. And you have to answer the political question. Whether it's scientifically valid or not doesn't really matter. This is very much a political question. You just have to do it. And you have to show that you did it. I know in the US, the definition by the National Library of Medicine of ethnicity in the US is very, very focused on congressional districts. It's basically defined by congressional districts because they use it for funding purposes. So it's a very complicated issue and it's a very complex issue, and often it doesn't matter if it's scientific or not, you just have to do it. And the only way to do it is collect from a broad base. Life in the big city. Could I ask you to go back a slide? I have a question on, or even 2 slides. One more. Is it this one? What's the difference between cortex and cortex of kidney?
That would be a DCP don't necessarily apply biological rationale. So we was that sample available when it came to their plans.
Okay. So you guys are having the same problem that we have, in that every biological concept has 4 official names and 19 unofficial names, and this is the kind of thing, well, that makes a big difference if you suck those 2 together. Suddenly that becomes your biggest group? No, not quite. It catches up almost to not provided.
I would argue not provided is probably cortex too, just by frequency of sampling of that site.
Do you have any, we're struggling with this right now, doing the kidney. We'll generate an ontology, fine. I mean, there's 500 ontologies out there. They don't necessarily agree. You can't aggregate them because then you do things that are downright incorrect when you aggregate ontologies. On the other hand, even when you get to a good ontology, you have to have some way to feed it back to the wet lab that's registering a sample, so they can say this is KTAO 47928, which has 7 different names, 2 of which are cortex and cortex of the kidney, and et cetera, et cetera, et cetera. So you're still struggling with that as well?
Hopefully with our samples that we've submitted, we've tried to just broadly say whether they're cortex, medulla, pelvis, ureter, and probably that's our ones that are coming out.
But you can't use cortex.
We can use cortex, yes. So
no. I mean, if you take cortex out of context, it could be the brain.
Sure, but they're obviously we're submitting them as part of.
So I mean, that's why cortex of the kidney is a little bit better because it's a little more specific.
Yeah.
If you add an ontology term to that, then there's a definition attached to it. And as long as the submitting entity knows what that definition is, then you get beyond that you accidentally not find in brain, collecting duct cells profiled. And this happens regularly if you don't do it. And so this is another dimension. To what extent do we want from the submitting entities, do we want to map against a standardized nomenclature? This gets particularly relevant for all of the clinical features, where if you don't do that, they also become not interoperable for that very reason.
I just wanted to say.
Can you also introduce yourself?
Yes. As a member of the DCP team, I'm a user researcher. So in my job, I actually go and get feedback from submitters and from everybody else on things that could be improved. But specifically in this case, I think the data wranglers are doing quite a lot at the moment it's all quite manual. So in communication with submitters, they're trying to go and put the right ontology. So they're communicating in this case. It might be that, yeah, it might be a matter of just this was really not provided and they couldn't get to the right one. But I think they're also working to actually have consistency across the different data sets and not have those like multiple options there. And of course in the future we will try to work towards some kind of automation, so even when somebody submits they have a way to actually go and select themselves. I think we're still looking a little bit into the future for the automation, but we're thinking along these lines.
Yeah, so I think that just as in the same way you can determine genetic similarity or differences, so we're talking about if you want to call it ethnic origin or whatever, in the same way you will be able to infer the exact anatomical location from the data that you get ultimately.
Unless you have 500 samples, 250 are brain cortex and 250 are kidney cortex.
Well, I think, yeah, I mean, I think that, yeah, but I think from that point of view, obviously I think there is data being provided that this is initially a kidney sample and then beyond that.
Hi, Sarian Bowers from the Sanger Institute. I just wanted to go back to the ethnic representation. So I think one of the best ways to improve representation in your samples is to improve representation in who's collecting and who's actually producing those samples. And that not only will get you a better diversity of samples, but it will also fulfill some of HCA's mission a bit better as well. So I think some focus should be thought about trying to recruit research groups in low and middle income countries particularly.
Well, our group tried to capture that in our initial, so you know we are getting tissue from a lot of different sites, so we've favored getting smaller numbers but from more sites. There are 4 contributing sites right now, and you know if we include other samples like you know anything that would be available from H3 Africa or any samples from Asia that will be available down the road, I think that will increase our ability to capture more diversity ethnically. But right now we have 3 US sites and 1 in the UK. So we're trying to build that in as much as we can. And also to the point of, you know, we're talking about ethnic representation, but just to the point of how different kidneys are, there's also a very strong sexual dimorphism in the kidney, especially in the proximal tubular compartment. And so it really, we should be looking for these differences, even if we think that we're genetically pretty identical. There's a lot of differences at the level of the tissue that we're learning about, just scratching the surface, I would say. So keeping an open mind to your point. But beyond trying to collect from multiple sites, yeah, I'm not sure what else we can do.
Yeah, and I think as the technologies get better, then we'll be able to do that better. So if samples can be shipped from different places, because I don't think the fact is in the current situation, in the current climate, the technologies that we're using are extremely expensive. And so I think you can't just magic up groups that are in low-income countries that are going to be able to do this. So I think as our ability to use samples from different places improves, and they will continue to improve, that will address that to some extent. And but I think as long as I think if we have an awareness that that's definitely an aspiration, which in some ways is why we put this as something we should discuss.
And I love the strategy the lung proposed, yeah? That you have these different stages, and we are probably transitioning from the pilot to the first training and test stage. So this will filter out technologies who carry the most information content. And then if they can be scaled down or up, depending how you want to define it, to be able to use formalin-fixed paraffin-embedded tissue, then you can go global. Or if it needs RNA later-fixed tissue, then you still have cohorts around the world who have that material available. And that might be one of the prioritizations we might want to consider in the next phase. which technology is actually scalable in a framework that we can have this type of global reach. There are formal and informal networks in place to actually access that type of material, but obviously not for single cell sequencing technologies.
I have actually a question regarding coming back to the annotation and also asking your opinion. I mean, there's a spatial component, and beyond an ontology, there would be again a gradient. Has anyone experience in having a part of the annotation being a 2 or 3 dimensional representation of the kidney, and you just kind of circle things where this location come from, and do you think that would be practical? Because I could imagine if, again, practically on the sample side, right, that might be easier than filling out, going through an ontology and all these terms, and maybe we underuse this tool, right? I don't know.
Sorry, and now we've got cross conversations going on, but I actually just wanted to slightly respond to your point that HCA is thinking about setting up an equity working group, and I actually think this is exactly the kind of thing that they should be working on, and so this group should be trying to feed back that that's the kind of thing, because I have a concern about that group, that it's actually just going to end up running in parallel to HCA itself, and so these really important questions aren't necessarily going to get addressed within HCA properly.
Didn't introduce me. I'm Jim Sluka. I'm from HubMap. One of the things that we're developing is a tool that lets you bring up a 3D vision of the organ and then to place a cube in it, or a rectangular pyramid, or a needle biopsy. So that you say, here's my kidney, and I'm slicing it up here. Inside of our kidney model, hopefully we can get help from the kidney people and get an object file that's got cortex and pelvis and such and such and such. So when you place your sample, it can automatically generate the annotations. This sample should contain cortex. It should contain this and this and this based on how it's aligned. It should not contain this and this based on how it's aligned. I think in the kidney, it'd be nice to know if it's the upper or lower pole or mid. It'd be nice to know exactly how you're going into the crescent shape. But beyond that, there's a little less detail. I think in something like the small intestine, though, it's going to be critical, because you're just going to want to say, I'm in the 3rd loop down. Instead of having to say that in words, just to be able to point at a picture and oh, by the way, my sample was taken this way relative to the body versus this way or this way. So we're working on that, and if we can get support from other groups, we can make it better and make it work even better, but we can demo it to HCA if HCA would be interested.
I think that would be interesting, and I mean, I guess one other way of doing it very simply, which we have done in the lab, I guess, in a slightly haphazard version, but is to just take a picture. So you've cut your kidney, because we have whole kidneys. So if you cut your kidney open, and we take a punch biopsy, so it's very obvious where it is. There's a big hole. And if you just take a photograph of that, it tells you exactly where that punch came from. So that's the other way of keeping the information.
If I can just sort of add to that point, Ben Stewart from Cambridge. The kidneys that we get don't always correspond macroanatomically to nice diagrams. So I think a picture mapped to a diagram is better than just sort of noting things on the diagram.
I think it's critical that the common coordinate system for HubMap, I think, might be of relevance for the kidney. We just don't know, because we never had that type of data. What is also important that we talk to our renal anatomist, and I had a PhD in that a while ago, that we understand that nephrons are not all the same, that depending where your nephron lies, that you have very different nephron architectures, and finding ways to capture that in structural term will be critical when we do the spatial mapping to actually ascertain where our different cell populations live. And that's an aspect where getting this domain expertise represented and hooked up to the molecular data sets we are currently generating, I think will be very important. And it was great to see from the presentation earlier this afternoon that with a nomenclature platform and framework, that actually is one of the options, hopefully, which will facilitate that. And in the kidney field, we hope to have such a similar meeting than the Brain Atlas had in July, in January, so that we can get renal anatomy, renal physiology talk to the cell biology which is generated here.
But I can't help but thinking, sort of goes back to one of the things that you said about every human kidney being different and what Ben says. So many of the kidneys that we get are a bit of a funny shape, or there's a whole lot of fat here, or there's a scar, or there's a... So actually it may be that even if we have a common framework or a 3D diagram that we can put where our biopsy is, that still it might be very valuable to have a photo that just says where it's from, that then is captured with that biopsy.
And the histology. So I think what is clearly important is, and I think Yul Bale is represented at the last meeting here, that we capture the structural features at high resolution and in a way that they are digitalized and can be used for automated image analysis. And because none of these kidneys is normal, certainly if you go at the histology scale, you clearly will see a wide spectrum of structural differences, and having them available to interrelate with the molecular information. We will be limited with sample size for a while with that, but certainly in a semi-quantitative way will be very informative.
Because that sort of touches on the thing of what is health and what is disease, and you know, I think especially as we sample across lifespan, then there may well be anatomical differences that develop with age, and who can call whether that's health or disease. It's
yeah. So we've been thinking about that. It would be great to have a operating room photograph. But that doesn't work very well for some tissues, right? Lung collapses, and taking a picture of it out of the body is not quite as useful. But still, if you've got that picture, you want that picture, and you want to scale on it. The other thing is we'd like to be able to do is, if we're working with OBJ files, to be able to use the patient's micro-CT. And longer term, being able, I mean, the same tool could be used for somebody that's doing a liver resection, and the surgeon wants to know where the major vessels are. And he doesn't want to just assume they're where everybody's major vessels are. He actually wants to look at it in some other way. So we've thought about operating room photographs, a standard reference organ, and a patient-specific micro-CT, and hopefully being able to feed at least 2 of those into the same tool that lets you line up your little, whatever your section is. And then the last thing we're trying to get in is, so can you do it as a needle biopsy, or especially in experimental animals in the liver, there's a standard way of slicing up a liver in a rat liver. So you just code that in as a standard so that somebody doesn't actually have to go through, well, we sliced it this way, and then we sliced it this way, and then this way, and this way. It's a standard procedure to have all of those built into the tool. And that's actually pretty easy to do. The tricky part is getting the original 3D models from each individual set of organ experts.
Cover those subjects around tissue collection, metadata?
Yeah, I think around this, because I think there's obviously a lot of interest in this capturing the spatial information, then maybe we could try and have an action point to take forward. So, I mean, you would...
HubMap, HCA, HubMap demo with HCA is another conference for the alignment tool.
Yeah, so that sounds...
If we go by the tetrahedron that was presented, that would be covering the position part, which I think is one of the things that we should probably, I mean, it might be the easiest one to align on, beyond, cell types and states. We're going to have a lot of debates to figure these things out iteratively, but position we should be able to somehow figure out more straightforward.
And I think then, you know, maybe it will be possible for us to make some recommendations of the ideal for samples, so that ideally there is information about where the sample was from, preferably a picture of where the biopsy was taken from, and that where possible a parallel sample would be processed for microscopy, so it, you know, so put paraffin embedded.
For KPMP, for the initial pilot study, we have developed a detailed manual of operations for that. We even have training videos, which I think are also publicly accessible. So if this is of value for this group, I think they are clearly shareable and could help to, again, define how you capture formalin fix, what's your formalin fixation time period. We even standardize different formalin fixation protocols among the participating centers and identified variables which can impact some of the digital analysis there. And this is, as long as this is under an experimental heading, which it will be here, and not piggybacked to a routine diagnostic procedure, something which should be implementable.
This is Yasser Yazal-Hosseini from McGill University, Canada. I was just not sure, because my understanding was that mainly the discussion is around covering different ethnic groups and samples from different areas. Was not sure if it is now focused only on non-diseased tissue, or you are also planning to include diseased tissues, in particular kidney cancer. Or is this something that at all it fits to this session and group, or do you have any plan for that?
Yeah, I mean, I guess ultimately we would want to cover different disease tissues, including cancers. I mean, I guess from our point of view, we've done some work with kidney cancers, principally because the source of at least some of our normal kidney sample is adjacent normal to tumour. So actually I think those studies lend themselves well to that. But I think ultimately, yeah, we would like to extend out into different disease states.
But I think it's fair to say that the explicit effort of the HCA was to really identify normal at the single cell level. And you know, our funding is around that at this time, so that's pretty clearly stated. Of course, it is very often the case that, you know, human tissue is obtained because there is a reason, usually, and it has to do with detection of disease or sort of diagnostic maneuvers for that, depending on the type of tissue. So it is understood that diseased tissue will be also available, perhaps at the same time, or, you know, under different circumstances, but our efforts here are supposed to be focused on the normal human. Oh, Ben, back to the same.
So perfect. I understand that, but mainly I'm interested in using the experience of different groups. If, for example, you're already working on kidney cancer, is it something that we can benefit from your experience? Because we are working as well. Maybe the tissues are not going to be included this project, but maybe this is a platform for building up more connections and discussions for future. And well, if, as you mentioned as well, we have access to the normal kidney from same patients. If that can help, we will be happy to answer.
One way to address that comes to my mind is to kind of jump to the last part of our list of questions and talk about sort of communication, collaboration within the team. And the team is a very kind of ill-defined entity. It's ever-growing, and it's kind of an open invitation for people to join. So maybe I'm biased because my lab uses Slack, but we have a Slack channel that is open. I can put up the Slack address so people can be, you know, invited or join in the Slack channel. It's kidneyhca.slack.com. It's very straightforward. And so the idea would be to use that maybe as a place to say, hey, is anyone else doing, I don't know what you're doing, like RCCs or whatever other tumors, you know, you might be interested in. And then maybe there would be someone, you know, in the group who's interested in that. So that might be one way to foster that. It's supposed to be a place where we are all, communicating about what's going on. And, I would argue that we could be doing better with that. We haven't really been very good about using the Slack channel as a community so far. Maybe this would be one of the action items from today to try to encourage each other to use it. I use it daily, just not in that particular channel. We haven't been sharing a lot, so I could definitely, adapt to that more readily, and I hope that maybe others could as well. I know not everyone uses Slack, but it does seem to be a good place for a community like this to go. So I don't know if I'm jumping subjects, but to some extent, maybe that invites us to talk about that part. Any other ideas about team communication or collaboration? We covered a little bit the interaction with the other groups, and it's sort of left to you guys to talk to HubMap and KPMP and see if there might be an appetite beyond the conference call to demo the registration, tool, that maybe there would be additional interactions that might be good to have. At least one meeting where we all come together potentially. I don't know if other groups, I don't know if anyone knows if other groups, other organ systems, so to speak, have had their own special meetings. Do you know?
So I certainly know about the fetal cell atlas. They certainly use the Slack quite liberally, and so more than
so
we
that
do.
could be a great way. I mean, I don't know, convincing the other groups like the Rebuilding a Kidney or the KPMP to join a Slack channel as well might be another way to really bring everyone together. And there are a lot of people who are in common between these different groups as well. So maybe that would be a way to start, as Ben is suggesting. Other thoughts or ideas?
I'm Mahmoud from RWTH Aachen University in Germany. Can I jump back to the? All right. So about the annotation of where the sample is and what it is and how long it stayed. I think it's really from an analysis point of view. If I'm analyzing the data and I see a difference, it's much better if I can know what this difference is from than not. And I saw the conversation going more towards making everything homogeneous, like having one figure that you can annotate the sample on and so on. But there might also be a case for allowing, additional to that, allowing free-form sort of annotation of every sample, that the scientist who's collecting the sample can just freely write what they did, and this can be later processed by text mining or something like that to come to some more formal thing. But it will help a lot, I think, in the future when the data analysis, when people actually bring the data together to understand what the differences that they might see comes from and what we need to be more accurate about. Maybe ischemia time is very important, but whether the sample is like this or like that is not so important. I don't know.
It's not one or the other, it has to be both. Whatever can be standardized needs to be standardized. The data wrangler will enforce that. I'm not one, but I'm a victim of data wranglers frequently. This is really important. When it can be standardized in the metadata fields, we have to use choices, drop-downs, so they are computable. And then you have to have the extra field and say, look, this patient bled during the nephrectomy and had a blood pressure of 80 for 25 minutes prior to the organ coming out, which there's no pre-specified field to capture that. And if you see a dropout of your proximal tubular epithelial cells, that might be just a consequence of these cells already being on their way to acute tubular necrosis and thereby not captured by your assays. So that's, I think, it has to be both. And again, that might not be needed to be reinvented for this network when other networks have some of or other organ systems have some of these metadata fields already in place. It is work for the tissue procurement labs to capture that information, but I think it is critical, and ideally you have a submission form where these fields need to be populated before you can actually upload and enter samples or data into the system. And it comes also back to your analytical output, if there is a standard framework to capture your analytical variables. And that's the same from, yes, that was the day where in Aachen we actually had 42 degrees Celsius and we don't have air conditioning. And that might be one of the main confounders why their data set looks different.
So I think, Ben, you've uploaded data to the DCP.
Yeah, that's right.
And I mean, they do have relatively rigorous metadata requirements, right?
Yes, and I think it would have been good.
Understatement.
And it's it would have been doing the study again. It would have been good to have seen those, because we would have probably collected a lot of that metadata. So it's great. It's great to know what we need going forward. And I think, Gabby, you probably could show people where all that information lives on the HCA portal.
Is it possible to post it on the Slack channel, for example, so that everyone can click on it and see?
At the moment, we're actually thinking a little bit more on how to engage better with the communities. And I think even like last week, there was a plan to launch these conference call where we can invite people so that they can give us like more feedback. There's also a plan to better represent it on the data portal. At the moment it's there, but it's not in like the possibly like the best possible way for people to actually understand it. So we're trying to make that clearer and to engage better with the different communities on different, the different fields, the different things required. Yeah.
I mean, specifically, so as I understand, the KPNP has a metadata framework, right, which would not differ terribly from the metadata framework needed, I guess. And I mean, concretely, has the DCP communicated with the KPNP? I mean, just out of curiosity.
Wait for a moment.
I'm not sure about that communication specifically. I do know that obviously with HubMap there's a lot of communication going on, and there is like a common call which happens every week, or I think regularly. So I think this communication is definitely there, and they're going to try more to actually collaborate.
It's actually kind of amazing that NIH has been funding biomedical research for 80 years. You would think they'd have a standard patient table. Body weight, height, they don't have it. Every time they fund every $5 million grant to study something in humans, they generate a new data table for them. So, so it sounds bad when they say, there's 3 groups here doing the same thing, and have they shared their metadata yet? The fact is, it should have been defined decades ago, and it hasn't, and the way it is always done is you generate a new one. So if we share, we can actually break the mold.
Exactly, that's the point. This is where these groups have a clear recognition that our data utility will be significantly impacted if we don't do that. And that cuts across the different networks, the different funders, and the different investigators. Yeah? And that overcomes some of the proprietary aspect of all of these, that there is so much to be gained. And all these activities are early on so that this can take place. And that's... Yeah, but, but this is besides the point. I think we want, as investigators, we have a clear charge, and we will make things happen as always. Yeah? Despite funding mechanisms. Sorry.
So if there's an existing phone call or conference call between HubMap and DCP right now, would it not make sense to add KPMP to that?
So this is the general HubMap.
I get it. So when you're talking kidney, maybe.
So exactly that's, I think.
Yeah. Of course.
Hi, I'm Zarina Gallus. I'm one of the leaders of HubMap. So one of the things that Jim suggested is a big failure of NIH is actually not a failure, it's by design. NIH is allowing investigators to come with proposals, and it's not imposing strong enough regulations, because I don't know about here, but in the US of A, nobody likes the government. Although all their salaries are paid by the government and all their research and so forth. So we were not allowed to impose specific regulations. So what happens, we announce the calls and people apply and the best applications are selected. It happens that some of them were kidneys, some were lung. We didn't tell them what they should be. KPMP, the leader of KPMP from the NIH side is also the leader on the kidney program in HubMap. He's the same program officer. So it's not that we are not talking to each other, it's actually the same person. We talk, she's also part of the HubMap working group. We have a working group with probably a dozen institutes. There is a meeting that's being planned, and you heard about it. I think it's March 30th, April 1st, where we're inviting everybody who has any interest in mapping any organ, any tissue, to come together. And there will be interest groups, and there will be sessions that are going to try, because we are all trying to conquer similar challenges. So please look at us as a partner. By design, because NIH is using taxpayer money, it's very difficult to give money outside the United States. So we realize that. So we're actually constantly working with CCI and the other funders who are able to fund across the world. So it's much harder for us, it's not impossible, but we have to demonstrate that anybody from outside the United States has something that nobody in the United States can produce, which is not that easy to demonstrate, right? So we are open to collaboration, so we are working within our confines of, so again, we are trying to impose the request that everybody shares their data, but in the end, it's up to investigators. So I totally encourage you to share everything you know. We are here because we want to share, and we want to know, and we want to work together to conquer the challenges. This was long, but I wanted to say a few things that I heard. And
no, that's brilliant. I mean, it's an excellent comment and very well taken. And it sounds like a solution to a lot of what we're discussing is, and I know that it was put up by Aviv and Sarah earlier today, this March 2020 meeting, which is kind of around the corner, it's not too far from now, could be a place where, you know, there could be a similar breakout session, except that maybe more stakeholders are there representing all the other groups.
The other thing that I didn't say is that Aviv and Sarah and a lot of people you see here are also funded by HubMap. So there is not an actual sort of disjunction, and a lot of the things you've seen were actually produced with money from HubMap or joint money and so forth. So we don't really worry so much about where the money is coming from if the knowledge is being generated and it's openly available to everyone.
Yeah, that's what I'm focusing on, to talk about sharing of our knowledge and maybe agreeing on some common parameters around, you know, specifically for kidney, what we're discussing, you know, position, you know, even annotations and other things that we haven't even gotten to yet that were discussed earlier today as well. It sounds like, you know, it's often the case that if you put all the stakeholders in the same room, there will be more ability to have consensus. And so it sounds like March would be the right time to really attack this problem,
and actually, as
or
you
start
heard,
to attack it.
No, actually, I would even say we shouldn't wait until March, because that's 5 months down the road. Having, you know, as we just heard, you know, a video where the common coordinate framework from HubMap is explained, to do that, to share the protocols. And again, I have to be careful. I speak here, I cannot speak for KPMP, but I can bring that message to KPMP, and I'm sure we will get endorsement and permission to bring these protocols, to bring the meta standard forward and continue the discussions. I know Becky stack from the DVC and KPMP has already initiated and has shared documents back and forth between human cell on the ground floor. That things are connected up there is great. We have to make sure on the ground floor the same is happening. And as we are developing and pushing protocols out here now, this is a time to make sure we coordinate wherever possible without stifling, obviously, creativity, that each side can leverage what they have available, but they don't need to reinvent. So setting up a follow-up, theme-specific contacts via emails, conference calls, I think will be an action item I would recommend we take forward. And then that might lead to the March-April meeting to have a kidney-specific sub-meeting at that time as well.
Yeah, I wanted just to say that if you are all interested, I thought that was a great idea to have sort of an organ-focused either group meeting then or deciding to have a meeting, whatever, steps. We obviously look at the same organ, so we should leverage each other's. So KPMP, because they were looking at development, or they were looking rebuilding the kidney, the disease, whatever. So we are doing the normal kidney, but obviously we learn from each other. And the reason we didn't do mouse is because we're told by Deborah that they actually had a very hard time taking the markers from the mouse to the human, so we decided let's not do that. Development has its own challenges, so we thought let's do the easiest thing, which is normal adult. So it's not as easy as you all know, but let's put sort of like a baseline if possible.
So speaking as somebody who sits on a lot of working group meetings, for HubMap, KPMP, Brain Initiative. So yeah, it would be nice because we're constantly tasked, I'm constantly tasked with the same job in those working groups. So it would be nice to just do it once across all of the sites, just have one common working group. And whatever has already been done, we could just easily fold in rather than starting from scratch and doing it all over again. Yeah, we have to do that, and then also just not reinventing the wheel and wasting our time, because it's so much time doing all of these things. It's painful, actually. So it'd be really nice if we could just take what some other group has, and then we could just add to it and just keep building, right?
Okay, so any more comments on that? So I think that's a good action point going forward. Okay, so if not, maybe we could pick up some of the other topics. So I think one thing of interest is how far we've got in terms of spatial technologies and the kidney. So my own experience is that human kidneys are extremely autofluorescent, and there are challenges with generating nice-looking data. If you're looking at immune cells in the kidney, which I'm interested in, so in normal, they're relatively sparse. So actually, for example, using RNA-scope, sometimes it's difficult to distinguish what is fairy dust versus what is real signal. So I'd be interested to know other people's experience of using spatial technologies of different sorts, whether that's transcriptomic or protein-based assays.
I could just speak to our efforts, which were shown this morning, and everyone can tell that they're not quite there yet. But we are working hard with Faye Chen and Jamie Marshall to get SlideSeq to work in human kidney. It's in development, I would say. We are starting to see some structures that make sense. So this SlideSeq is basically a technology that's based completely on transcriptomics in terms of being able to generate an image of all the kidney cells spatially arranged with each other, just simply based on their transcriptomes. Faye Chen and Evan McCosco developed that, and it works pretty well for brain, but still kind of in development for kidney, I would say, is the update there. There were some people here from BioLegend who left, I think. They are developing an entire panel of antibodies specifically for kidney based on transcriptomically derived markers from all of our work. They're looking at all of our different, papers and trying to develop markers. And it would be great to give us an update. The last that I heard from them was that they have about, they have the ability to do 200 at a time by specifically, titrating antibodies and taking care of other technical details that allow them to do this. And so I think that's pretty exciting if we can get ultimately a panel that works well for landmarks in kidney. But that's the extent of what I know about that. So any other methodologies for spatial? Murfish? Any of the others? Anyone? Jamie Marshall is also working at the Broad on HCR, which is another in situ method for localizing several probes to kidney, and it works pretty well as a kind of validation tool for a lot of the transcriptomic work that's going on. But anything else on the spatial side?
I mean, not that I know anything about it, but I mean, as you know, in this STVG proposal for benchmarking the different methods, the spatial methods have a big role, right? I mean, we have 5 kidneys from both of your places, right? And then there's Codex and there's Murfish planned, but that's all planned. First, we have to get the tissues.
And you have to tell the procurement sites how you want the tissues.
Yes, definitely.
Which maybe, I mean, if there are no comments on that, I mean, I think in some ways the fact that there aren't so many comments reflects how difficult it is to get good data. But maybe related to that, obviously the bulk of tissue samples that we all have stored are formalin fixed, paraffin embedded tissues, and any updates on being able to generate data from these. I don't know if anyone's got any updates in that regard. No, so right.
That's a tough one.
Yeah, it is a tough one, but I think if it can be resolved, clearly that is where we all have the sample sets stored. But anyway. Yeah. So in terms of other technologies, so single cell ATAC-seq, so experiences there with the kidney. So I know I was at the European Society of Transplantation meeting and organised a session on single cell omics, and Ben Humphreys came and presented some data. So they've done at least one sample using single cell ATAC-seq, so started to develop that. Anyone else with any experience in that regard? We ourselves have started that on one sample set, but we haven't got any reasonable data that I can tell you about yet, but we're starting on that journey. Okay, and then any thoughts about rare cell types in the kidney and strategies to identify these? So I guess from our point of view, immune cells are already in the minority. So then if we're thinking about subsets of immune cells, then definitely this is a problem we're trying to address. So as we heard, I think it's from Orit, so one way of going after this is to try and enrich for at least broadly the cell subset that you're interested in. So we've been interested in mononucleophagocytes, so trying to have some sort of selection strategy or sorting strategy. One of the problems that we've found anyhow in terms of mononucleophagocytes or macrophages is that by the time you digest them to get them out of the tissue, that they become pretty unhappy and die, and so actually getting good data is problematic. Again, I don't know if anyone else has any experience. I mean, I think particularly when you're trying to get samples out of the medullary and pelvic regions, because the fibrotic or the infrastructure of the kidney is just much more difficult. You know, by the time you then get them out. Yeah. So Neil, yes, Neil is a hepatologist who's moonlighting in the kidney session.
Yeah, I'm an interloper, sorry. I'm a liver person, but it's been a very interesting discussion. So, I mean, the rare cell type thing, do you think, you know how with nuke-seq from frozen tissue, you should be able to drag down pretty much most nuclei, but you can be dominated, presumably in the kidney, by epithelial cell nuclei, etc. But do you think that's a possibility for going after rare cell types, or do you think the transcriptomic depth is just too shallow to allow good enough annotation?
Yes, I think that's definitely one strategy. So that's something else that we're trying to pursue, and but having some sort of step where we select out because the bulk of what you get out is proximal tubular cells. Mm-hmm. So if you have some kind of strategy just to get rid of all of those, even then you're in a better starting point.
Yeah.
If I can say something about the single nuclear RNA-seq, so it might work better for getting all the cell types, but the problem is with the 10x platform, you select for poly-A-RNA, and this is really not the most abundant RNA type in the nucleus. So there has been, I don't remember now who published this, but there's been a couple of protocols that actually do single cell RNA in the nucleus, so nascent RNA, and this would, if this can be coupled with nuclei isolation, the dissociation protocols that we follow in the kidney, then this would be maybe a more appropriate way to assay RNA than the 3' poly A 10x assay for single cell nuke-seq. Just.
Can I say, in terms of rare cell types that I don't think I've seen in our dataset or in any other published datasets, I think chiefly stromal cells, fibroblasts, myofibroblasts are missing, but then also some minority epithelial cell types, so particularly distal convoluted tubule and juxtaglomerular apparatus seem to be sort of conspicuously absent, and I don't know whether anyone has found these cells. Anyone have an experience?
So we've been getting higher sampling by changing our assay, and so now we can start seeing a lot of these rare cell types. But with nuclear-seq, I'm a Blue Lake from HubMap and etc. But with nuclear, you do have some drawbacks in that you don't get as many immune cells. Maybe the type of nucleus and the amount of transcripts in that nucleus will be a big factor. So immune cell types tend to be a lot rarer in that context. Also, I wonder if anybody else is starting to detect neuronal cells in the kidney, if that's something that people are seeing. But
wouldn't just the end of the neuron be in the kidney in a way, or?
Yeah, Ben, do you want to say?
So we've, in our fetal kidney data set, we have some neuronal cells. It's likely that, it's a very small structure, and you take the whole kidney, and we just nicked a bit of ganglion. So it's possible to get the cell body of the neuron. That's as much experience as we have, I think.
I don't know anything about neurons in the kidney, but I mean, what I do think and also see in other areas, especially of the brain, is that nuke-seq is getting more and more a standard. I mean, there are difficulties, but the practicalities of getting the cells at all or the nuclei at all is just so much stronger. I mean, we could have a perfect reference if we just are able to do that once. it doesn't help, right? So I do think that nuke-seq is getting more and more a standard, although it's kind of counterintuitive what you should get out, but the poly A priming is also not so specific in the end, and yeah, and maybe one would even come up with better ideas eventually. What I do think could be extremely helpful to link that to the rare cell types. In principle, it should be possible to get antibodies or maybe smaller structure like nanobodies into the nucleus without destroying it. So what is something that is not at all developed but could be very interesting is to combine nuke-seq with staining of markers, because that's really lacking. And that would allow then with particular protocols, because I mean, one thinks the nuclei are the same, but they're probably not, right? I mean, they're just as different. I mean, they're also very different. I think that could shift, I mean, that would make a lot of things possible that are now really not at all possible. I mean, from frozen samples and less bias, and then selecting even nuclei, I think that would be a road that could be productive, but I have no clue.
I would caution to, trade one bias against the other. If we have an opportunity to capture with two parallel strategies from particularly the same material, then you very quickly will see that they are complementary. And this is what KPMP we have done with a bunch of tissues, and you clearly could see that each of these techniques brings or enhances the visibility of a population which you don't see in the others by whatever underlying biases you have, and it also helps you to learn where your blind spots are with one versus the other technologies. But obviously with single cell and sorting single cell populations out of the kidney, the AMP in lupus nephritis has done that quite successfully and enriched for unique cell populations. But the main concern is, as you stated, you know what you find and you don't know what you don't find, and that's where the spatial technologies are so important, that we can close the blind spots. And with neurons you can pick up neuronal transcripts in peripheral nerve fiber, even if you don't get the cell body. So this is why these technologies will be, for the kidney more than many other organs, absolutely critical to come forward. So the task is, with the groups who are developing these technologies, if you get a hunch how tissue needs to be preserved for these technologies to work best, really share that early, so that when we are harvesting tissue, that tissue can be evaluated, particularly if single cell data sets are available from the same material already.
Yeah. So I think I'd make a comment on the you don't know what you don't see. We know from in mouse immunology with kidneys and other organs, if you and if you take if you take a mouse kidney, it's easy to mash up. It's easy to generate single cell suspensions. But even then, your estimate of the immune cells that are present in there, so when, if you just take the organ, slice it up and actually laboriously count the immune cells of interest, the FACS, underestimates it by 100 times. So whether those die or they're just not released at all. So I think we do need to develop the spatial technologies in parallel. There was one other thing that struck me just based on our own experiences. I don't know if anyone has tried, and it was another thing that I think Orit or Raviv mentioned. I don't know if anyone has tried cell hashing on samples that are not PBMCs, because we've found that maybe it doesn't quite as cleanly identify different samples as the data that's presented as the benchmarking data. I don't know if anyone else has found that with using actually tissues rather than PBMCs. Has anyone else tried cell hashing kidney samples? Yeah. I mean, we see a substantial population that just don't seem to be labelled, or even labelled with two different cell hashes. And So it may not be as straightforward as presented for blood, as with many things. Yeah.
Can I just offer a suggestion on that? So the protocols that have been devised for cell hashing in blood use antibodies against the sodium potassium ATPase and also beta-2 microglobulin. And I think that's probably going to be a problem with kidney epithelial cells, which might express variable levels of sodium potassium ATPase at their base lateral membranes. So it's worth thinking about.
But you probably have not tried to mix different patients and then deconvolute by SNPs, or because that's practically difficult, or because I think that's...
No, we can certainly do that, but if you're, for example, trying to follow something longitudinally, or you have a sample of the cortex and medulla that you want to run side by side from the same individual. So yeah, I mean, we've if we've got individuals that are genetically different, then yeah, there are ways you can go at that.
I mean, I think what might be a potential good alternative is this multi-seq approach, which uses lipids and a barcode, so then you do not have the problems of that the antibodies are present, and then you just needs to integrate into a membrane, and that could potentially be more flexible with nuclei and with cells, and independent of the antibody. And so, I mean,
is that something you've tried and got experience of?
Well, we tried it once essentially, but yeah, I mean, we're just very small. But I mean, the method itself is rather convincing. They also send out freely their barcodes and these lipid moieties until they've founded their company, I guess. So rather hurry up, I guess. Yeah. But I mean, just in principle, to have a lipid that inserts into a membrane that has attached a barcode that can hash seems to me a more promising idea than to choose an antibody where you do not know which antibody to choose, etc.
Yeah, and expression levels may vary. Yeah, no, that sounds great and very interesting. So maybe I'll try and get hold of them. Any other thoughts on that front?
So back to the rare cell types, and maybe, you know, when we're doing kidney, we get a lot of proximal tubules, and in the brain, we also can get a lot of layer 2-3 neurons, for instance, or oligodendrocytes. So one of the common practices in the brain, number of sites used, is transcription factors in the nucleus that you can stain and sort, select against, and reduce that. And we've used neuronal antigens to separate out the neurons from the non-neurons and then balance populations so you don't have oversampling. So it just involves finding the right antibody for non-fixed nuclei and working out all the sorting parameters, but easy, right?
Yeah, thanks. Yeah, that's a useful tip, but might need some working up. Okay, so I think we've probably covered most things. Is there anything else that anyone would like to talk about or feel would be useful to cover?
One last thing, because I'm just not aware how that is. So one problem, coming back to this annoying first part, the legal part, how is it with informed consents on different data types? Because what we had in this benchmarking study, right, that we want to do multi-omics, right, but then the informed consent was only for RNA and not for DNA. And are there some solutions that would fit, I mean, not only RNA-seq, but then also later multi-omics data together with the open data sharing policy with the HCA? And because in addition to the legal things between the institutions and the material transfer, I think that I see as a major problem currently that allows not to share samples and not to talk about even getting into the DCP.
I mean, I think that the limitation is really just the DNA. All the other omics, like if you would think about, proteomics or any other omic, lipidomic or anything else that you could think of on those samples, I think would be possible. The problem is with the DNA because of identification of the donor potentially. And so the IRBs are very protective of that. And I think we're trying to find ways to work around that. The main issue is making the DNA data open as well. because then that individual runs the risk of being identifiable, and the IRB wants to make sure that either that individual knows that they can be identified and fully understands that, and maybe there's one way to address it that way, or, you know, for some IRBs, that's just a kind of a complete no, we can't have that be open, it's too much of a risk. It depends on different philosophies on that front, but that's the problem with the DNA. All the other omics should be totally fine on these samples.
Just one, I mean, some of the DNA methods probably generate not much more genetic variation than the RNA-seq. So, I mean, from the logic of it, it doesn't make much sense, right? I mean, if you cover 0.1% of the methylome, then you're as identifiable as with the RNA-seq data, I guess. But
that's the point with some of the IRBs are now having the same constraints which are holding for DNA sequencing are holding for RNA sequencing, because the fidelity is high enough that you can identify individuals. And then the question is now, are you reporting read counts and not read sequences? And, you know, there are some solutions one can imagine to address that concern. I think this is good to see that there is an ethics working group in HCA, and I think this is not specific to kidney, and I really would look for guidance from this group, and they have been in these areas for many years, if not decades, and are leading many of the IRB discussions, and hopefully they can help us to come forward with information which we then can take to various IRBs at the institutions when they come back with questions.
But I think in the final analysis, we'd like to resolve this in a way that would allow us to share and, you know, for the future. It's just that it's not so straightforward, and we obviously want to be very serious about protecting the patients or the people who are, you know, donating their samples for research. So it's a balance that we have to strike and get it right. And it's rather important that we get it right now so that it doesn't taint, you know, any future efforts in this space. So I agree that having an ethics group as a partner is important, and this is not a unique problem to the kidney. Obviously, it applies to everything that the HCA does. So I know that there's a lot of thoughtful discourse around that.
Yeah. And I think it becomes more and more difficult as we better understand polygenic disease and how that plays out in terms of susceptibility, because knowing that genetic information will have implications, say, for people's health insurance, etc., etc.
I guess I just wanted to end again with the collaboration and communication piece, because I think that when we leave this room, that might be the most important thing for us to be able to carry forward. So I know that I pushed the Slack idea, but is there another way that you would like to see us communicating? We haven't been very good, so just to start from there and say, like, how can we do better? We already identified the March meeting as a touch point for interacting with other groups as well as amongst ourselves, and sometimes amongst ourselves anyway means multiple groups because many are represented in this room already, but what else could we be doing? Does it make sense to have some sort of email list and send emails? I hate emails personally, but, you know, everybody hates emails. Okay, I get a lot of nods, but what other means would you imagine? I mean, would it be sensible to set up a conference call that's sort of on a fixed basis? That seems very rigid as well, but it would force us to communicate. If we had like a quarterly phone call, I don't know, I'm just spitballing.
Because I guess the key thing is, what do we want to communicate? Right. So what are the things that's useful for us to communicate intermittently? And you know, because it is useful hearing how other people's experiences just here, and we have these meetings that force us to do that.
Every 6 months, yeah.
Yeah, but it's whether it would be helpful to have something in the interim.
Yeah. And is that satisfied by more of us adopting the habit of a Slack channel, or is it better satisfied by, I don't know, every 3 months conference call, or conference call is needed, or email, or honestly, I don't know what else to come up with, but I mean, I'll just, you know, post the Slack channel and try to see if people will start using that more. It'd be great if DCP will post something there, because maybe then people will go to that, because they will say, oh, what metadata do I need? Like maybe people will be more curious. And
I mean, you know best, because you're busy doing research. You know best what's the best way for you guys to communicate. But yes, I think DCP it will be helpful for us to be able to communicate with the group better. So whenever we have something around, okay, we need some evaluation on what we're working on at the moment in terms of, let's say, metadata, et cetera, and we want to kind of engage with that group, we will literally follow you. And yeah, a Slack channel would be great for us if we can also like get in touch with this community.
A website where documents can be posted, so if somebody is in the periphery of the group and wants to see what is happening, and then to attract them to Slack channel or whatever communication tools we are using. So that's, you know, kind of PowerPoints we are here, bullet points of the action items, the fact that we will have the follow-up meeting.
we could post the Slack channel to the kidney part of the HCA website. I don't think it's there at the moment, so that it would be easy to do. Yeah, could certainly do that. And yeah, we'll flash it on the screen today, maybe, and try to.
Before there was Slack, there was also online forums, which, you know, it needs to be set up and so on, but it's also, I mean, whatever, it's another option if Slack is too casual and you cannot search for things or so on, then
Totally
I mean,
search for things. I love it.
Yeah, sure.
And I don't endorse, it's not because I endorse Slack per se, I just.
Yeah, it's just another way that people were communicating and still are, is online forums, and if it's set up, then on the HCA website, for example.
There's a comment.
Hello, Norbert Tavares from Chen Zuckerberg Initiative. So I want to endorse the quarterly conference call, because you've already gotten an email from me, and I haven't heard back about that yet between the seed network groups, so at least there, and then more broadly. But also this idea about the groups, broadly kidney groups coming together and having a face-to-face meeting. If you can organize that and figure it out, reach out to a funder afterwards, once a plan is in place.
Brilliant.
Probably, possibly, there may be help with that, but
yeah,
because it sounds like a good idea.
It's good to hear.
Another thing I wanted to just throw out to the group, so this idea in the beginning about standards. So if actually a set of standards and protocols were generated that were benchmarked, would you actually follow them? So would you, in your labs, here's a set of standards, now change your procedures and go forward with these set of benchmarks truly, or is this just talk and bluster?
So I can speak from part of my lab. Yeah, I would if there was evidence there that it produced better data. I would have no problem with doing that.
Yeah.
So
if you have a problem, you would. If you don't, you wouldn't. Yeah. Or?
Yeah. What happens quickly if you don't and you end up with an observation which you cannot replicate, then the incentives increases very rapidly. So the experience was we shared very early the simple RNA later protocol to harvest tissue biopsies in 2003. And now this is a dominant protocol to use biopsy cores if you want to do transcriptional analysis as of whatever will we develop here over the next 2 years, I suppose. And that means groups who are adopting their technique with sample sizes of an N of 20 can link their data to several hundreds, if not 1,000 data sets out there and gain. And that, I think, is a philosophy many in the lab who have mixed background, not only wet bench but also dry bench, get, and therefore are willing to endorse. Assuming, and that's why the central data repository is so critical, assuming the core data set is readily accessible to you. And that's, I think, one of the key philosophies of Human Cell Atlas, to really build that attractor.
Yeah, so I mean, going back to the sharing also, one thing I think there's no alternatives to Slack, because everything else would just not happen, and if Slack doesn't get populated, that means there's not enough need to do it, and that's then okay, you know, I mean, that's how it is. And quarterly calls would be good, but I think we would need to find agendas where we really have a clear item and interest on what that's about. And I mean, metadata would be one thing, because I mean, yeah, it's bugging people for decades, and it's a community problem, right? I mean, that's nothing you gain much from it, and it annoys everybody. That's why it doesn't happen. And but now in this context, we need to get it working. And the last thing I think for sharing that should be on there is, I do think that protocols.io is really an orthogonal way of sharing and communicating the nitty-gritty details you would not get from Slack. I think I really would endorse that things are out there, because there people can take care about their protocols, and then if you have particular questions, right, I do kidney cancer now, what did you learn, right, then you can dive into there, and that's a different type of communication. So I think Slack and protocols.io and then dedicated calls would be the solution to group communication, and everything else will not happen.
Yeah. I take Norbert's call. We're going to try to organize these calls and you know, start to coalesce further. And I think it's time to wrap up our meeting. And I think the promise then is call will be coming up, and I assume there's a way to capture everyone who's in this room, because they would be participants in this meeting. So we'll be able to capture who was here, so we can include all of you. Certainly the seed networks will be involved in that, but others as well. We'll try to continue to use Slack. We have the March 2020 meeting as another touch point, with a kidney subgroup meeting potentially, and we'll go from there. Thank you all very much.
Yeah, thanks very much.